Hello everyone, my name is Kirtan and welcome to my channel Physiology at Once. Today I will be discussing about the part 2 of the topic Physiology of Immunity. The part 1 is discussed in the previous video. So let's start. Specific defense mechanism, which is also called as acquired immunity. It is the type of defense mechanism which is activated by specific antigens. It can be further divided into cellular immunity and humoral immunity. So let's talk about cellular immunity now. It is mediated mainly by the T cells. It is specifically effective against the intracellular organisms like viruses, parasites, fungi, cancer cells, tumor cells, and many more. The antigen is recognized and processed by the APCs, which are nothing but the antigen presenting cells, and then presented to the T cells through the receptors, which are activated and proliferated to destroy the antigens. The steps of cellular immunity are antigen recognition, processing and presentation, activation and proliferation of T cells, and elimination of the invader. So let's talk about the steps one by one. Antigen recognition, processing and presentation. The cells which process and present the antigens to the T cells are called antigen presenting cells or APCs as discussed now. These mainly include macrophages, dendritic cells and B cells. So now let's understand the major steps of antigen presentation. First, the antigen comes towards the APC. It is phagocytized. After the phagocytosis of the antigen by the APC, it is disarmed or there is partial digestion of this antigen in the vesicle of the APC. In the same time, there is formation of MHC2 molecule in the other side of the same APC. Then there is fusion of both the vesicles, and the next step, they both are fused together and form a single vesicle, and in the last step they are incorporated in the cell membrane of the APC by the exocytosis. So let's revise. It's the phagocytosis of antigen by the APC, partial digestion of antigen in the vesicle, synthesis of MHC2 molecule in the other side of the same APC. Then there is fusion of both the vesicles. Then there is a formation of common vesicle containing antigen fragments attached to the MHC molecules. These both attach together. Then there is incorporation of MHC Ag complex on the membrane of the APC. MHC is the full form is major histocompatibility complex. So now let's talk about the second step of the cellular immunity. So before talking about the second step of cellular immunity, first let's talk about the types of T cells. The types of T cells are helper T cells or T4 cells, cytotoxic T cells or T8 cells, and memory T cells. The first one are helper T cells. These are also called T4 cells because they contain CD4 protein. They assist in both cellular and humoral immunity. The helper cells which assist in the cellular immunity are type 1 helper cells, and the helper cells which assist in the humoral immunity are type 2 helper cells. Type 1 helper cells release the gamma interferon and interleukins, and the type 2 helper cells release the interleukin 4, interleukin 5, interleukin 6 and interleukin 10. These release the gamma interferons. The next one are cytotoxic T cells. These are also called T8 cells as they contain CD8 protein on their membrane. They are called cytotoxic because they kill the invaders by the process of cytotoxicity. They are also called as suppressor T cells because they suppress the antigens. Memory T cells. They can be either CD4 or CD8 dependent on the antigen. These remember the previous immunologic encounter, and on subsequent exposure to the same challenges, they are immediately activated without the help of APC. They have a very long life in the body. So now let's talk about the second step of the cellular immunity. Activation and proliferation of the T cells. The activation. The T cell receptors come in contact with the MHC antigen complex of the APC through the immunologic synapse, and then the T cells are activated. Proliferation and differentiation of the T cells. The activated T cells with the co-stimulation by the type 1 helper cells undergo differentiation into two sets, cytotoxic T cell and memory T cells. The next step is elimination of the invader. The killer T cells or the CD8 cells kill the microbe by three major mechanisms. Those are cytolysis. In this process, there is production of perforins which attaches to the membrane of the antigen and causes osmotic lysis and kills the antigen. Lymphotoxin release. In this process, there is release of many lymphotoxins such as tumor necrosis factor B, beta, which causes the killing of microbes. Interferons. There is release of many interferons, mainly the gamma interferons, which assist in the killing of the microbes. Now let's talk about the humoral immunity. It is mediated by the antibodies. These antibodies are produced by plasma cells, and these plasma cells are in turn formed in the B cells. The APCs are not a must in the activation of B cells. Steps of humoral immunity are presentation of antigen, activation of B cells, differentiation of B cells into plasma cells, proliferation of plasma cells and antibody production, killing of invaders by antibodies that include activation of complement system, which I will be talking in this video, formation of memory B cells and subsequent immunologic response. So now let's talk about the steps one by one. Presentation of antigen. APC or antigen presenting cell in humoral immunity include B cells themselves. So it is not a must for the induction of humoral immunity, but APC can also stimulate or present the antigen to the B cells. Then there is activation of B cells. The process of activation is accentuated by the co-stimulation from the type 2 helper cells. These activated B cells form the plasma cells and the memory B cells. The next step is differentiation of B cells into the plasma cells. The activated B cell enlarges in size that undergoes complete transformation to become the plasma cell. Plasma cell is characterized by the enlarged cytoplasm and more number of rough endoplasmic reticulums. The enlarged B cells appears almost like a lymphoblast and also called as B immunoblasts. The next step is proliferation of plasma cells and antibody production. A specific antigen stimulates activation of specific B cells that undergo proliferation of a selective clone of plasma cell. This is called clonal selection. These plasma cells secrete a large quantity of antibodies. So before going to the next step of the humoral immunity system, let's talk about the complement system. This system contains group of plasma protein designated as complement proteins. These are called as complement proteins because they complement the action of antibodies in killing the microbes. There are more than 30 complement proteins, but only 11 are categorized in this system. They are C1 to C9. C1 is again divided into C1q, C1r and C1s. They are normally inactive, but once activated, they exert their immunological and inflammatory actions and boost the process of humoral immunity. The next step is killing of invaders. Neutralization of antigen. The antigen is neutralized. Immobilization of microbes. The antigen is made immobile so that it can never further spread again. Activation of complement system. There is the activation of complement system by the classical pathway and C1 is the more stimulated one. The precipitation of antigens. The antigens are present in the soluble form. It is converted into the insoluble form so that it does not spread more. Facilitation of phagocytosis. The phagocytosis is facilitated through which the microbes are killed, providing immunity to the newborns. The IgG, which crosses the placenta from the mother to the baby, provides the immunity to the fetus. And the IgA, which transfers through the mother's milk to the baby, provides the immunity to it. The next step is formation of memory B cells and subsequent immunological responses. There is a small group of activated B cells which differentiates into memory B cells. They respond quickly and vigorously to the same antigens on subsequent exposure. This is also called as immunologic response. There are two types of humoral immune responses: primary response and secondary response. Let's talk about their differences. In the primary response, the antibodies are produced slowly. The latent period is 4 days to 4 weeks. In the secondary response, antibodies are formed rapidly. Concentration of antibodies rises gradually and attains a smaller peak. But in the secondary response, the concentration of antibody rises fast and reaches a greater peak. The antibody concentration returns back to the normal within few days to few weeks in the primary response. But in the secondary response, the antibody concentration falls very slowly and never returns to the normal range. The antibodies are mainly of IgM type in the primary response, and in the antibodies in the secondary response are mainly of IgG type. So that's it in this today's video. Please like, share and comment if you found this video helpful, and also subscribe for more useful and informative content about physiology. Thank you.